Okay, good morning, everybody. We are about to start this session. I don't want to take away time from the speakers, so I'll just make a few introductory remarks. Some of you may have been wondering what is the title of the symposium, Evolutionary Ecology in the Wild. And I've been getting some peculiar remarks from various people. Are this like wild ecologists? Ecologists behaving wild? What's going on? It actually comes from the title of a book written many years ago by a very eminent evolutionary biologist, and the title of the book was Natural Selection in the Wild. Now again, you may ask me, why natural selection in the wild? And the reason for that was that most evolutionary biologists have studied experimental evolution with Drosophila or C. elegans or all the other model laboratory organisms. Yes? So here we have a group of 4 very talented people who will take you through their systems or with ideas about how evolutionary ecology is really makes sense when you're looking at natural patterns that are occurring in nature. So without any further ado, I'll invite Uma Ramakrishnan, she's a fellow of the Academy and from NCBS. Uma, the floor is yours.
So good morning, everyone. I'm Uma, and I'm going to start. This is a new talk, so hopefully it'll be great to hear what you think about it. So as you all know from our high school textbooks, India and the Indian subcontinent has had a really interesting past. As an island, it made the longest journey of any landmass across the oceans until it hit Asia several millions of years ago. At the same time, recent data, this is a meta-analysis by Allen and others, shows us that basically if you look at the predicted risk of zoonoses or spillover of infections from animals to humans, India appears to be a hotspot, a potential place where a lot of zoonotic spillover, a lot of pandemics potentially like we had just recently, COVID-19, will happen. So I'm going to try and connect these two kind of facets of science. How do we actually study this? What are the consequences of the movement of the Indian plate on Indian biodiversity? And how does that then result in what we potentially see or what might happen in the future? This is basically a field of, biogeography, and I'll try and connect that to zoonoses towards the end, and hopefully you'll see a link. That's how I got to studying disease ecology and zoonoses that I do more of today. So I'm going to focus on small mammals. Mammals are our closest relatives, and half of all mammals are small mammals. In this case, I'm going to talk about rats and mice, old world rats and mice, and some shrews. So basically, biogeography is very simple, and it's a very old form of natural history where people like Darwin and many others, Wallace, Humboldt, why species are in specific places, which species are where and why. We can also think of this from a perspective of communities. Once a species is in a location, who else is around it? What species do they interact with and compete with potentially, right? Disease ecology is, you have all these species in a community, and then there are microbes laid on top of that, right? So for the microbes, the community of, say, rats and mice is their habitat, and they kind of move between species. For example, that's host switching or potentially spillover if the species in question is, say, a human, right? So basically, I'll take you through a few studies here, and then talk in the end about how, in the future, we hope to look much more specifically at zoonoses. Okay, so just zooming out, when we talk about which species are where, let's take a context check. We are in India. That's over here. And I'm sure you hear in the paper all the time, India has high biodiversity, India has high biodiversity. Well, for mammals, not really so. Southeast Asia, in our neighborhood, has much higher biodiversity. But, of course, there are some locations, for example, the Northeast has really high biodiversity. That's the comet that you see kind of going into India is the Himalayas, right? So basically, it seems like, India has reasonably high biodiversity, but of course, its neighboring areas seem to have more. So can we deconstruct this pattern? Of course, it has something to do with India's past. The fact that when mammals were speciating globally, India was an island floating through the ocean, kind of isolated from all of these processes. So if we look at that from a more quantitative perspective, what we can think of is that maybe India is simply a sink with species after it hit Asia coming in from the west, the north, and the east. And so we tested this with patterns, just patterns. This is called a macroecological approach. We just say grid up the whole of India, look at species in these grids, and plot these grids as a function of, where are most of the species, say, in a genus from. And it's really beautiful. It's like India is a blotting paper, and you have ink coming in from each side. And as you get farther from the sources, that ink or the number of species dilutes. So you can see here, for example, this is Western taxa, and you can see it's highest in the west. This is Eastern taxa, highest in the east. Note that the numbers are much higher. Sorry, it's getting cut off, but this is like much higher. The red is much higher here. And this is Northern taxa. The Himalaya, like a step function, they just don't get beyond the Himalaya at all. So does this mean that there's no, there's very little kind of endemic diversity in India? Is everything in India we have somewhere from outside? So how do we test this? Well, what we've done is we've actually put together a phylogeny. This took several years, actually a decade or so. And we did it for a speciose group. When you want to look at a process, you want to look at it with many species. And we did it with these guys. They're murines, so basically old world rats and mice. And there's about 704 of them, 704 murines in the world. And we built a phylogeny with 437 murines, so 437 of these. And this is the largest phylogeny so far. This is not published yet. But basically, the last phylogeny had only about 268. Of these, 40 species are in India, and we have sampled 34 of those. So over time, the lab has been working in various habitats, and we've been able to sample a significant amount of murine diversity. And what does such a phylogeny tell us? We can actually build this phylogeny and then basically ask, so that's the phylogeny. It's really messy because obviously it has so many taxa, right? It's like very, very long. You can't see much. But hopefully you can see these colored like nodes, and the colored nodes are what is called ancestral reconstruction of these areas. So the question we ask is the tips, which are the Indian species, where are they from, right? And what we find is, as expected, so basically this grayish blue is Southeast Asia, the big kind of dark blue is the Palearctic, and so on. And you can see basically that kind of as expected, sorry, this seems to have moved a bit. Most species are Southeast Asian, about 60% or so of Indian species are from Southeast Asia, right? So in terms of origins, species have come into India. There has not been a lot of diversification or speciation within India, and that is that also the phylogeny shows us. In fact, there have been more back radiations, species going back from India into Southeast Asia than there have been endemic species radiations. There's many questions that remain. Basically, did this happen at some specific points in time when the climate was conducive, wetter, so on and so forth? But this is something which we're still investigating. Okay, so that's fine. But I told you that I'm talking about communities, right? So this is some work which we completed recently, and it's looking at communities and disease in the context of the Western Ghats. I say right off the bat, this is potential pathogens. We have not tested pathogenicity of these microbes at all. What we did is, since we now know, that most species are potentially from Southeast Asia in a community, we looked at a biodiversity hotspot, the Western Ghats. We sampled 2 sites, Kadumane, which is a recovering plantation, tea plantation, and Kudremukh, which is a protected area for small mammals. How do we do that? We basically have traps, these little traps, where we bait them with butter, so the small mammals are attracted to them for food. but the traps could be up in the trees, as you can see here, or on the ground, and we set them up in a grid, and basically we catch animals, and you can see how many ectoparasites this animal has. Can you see all the blood spots on its ear? So these animals are living with many, many ectoparasites, which we basically sample both the ectoparasites and the small mammals to look at what microbes they co-occur with, right? So we basically can do some measurements, tissue samples, blood samples, oral swabs, rectal swabs, and look at endoparasites. And basically the methods are pretty simple, standard. We get these samples, both ectoparasites, sometimes if animals die, sacrificial, we take spleen, and then we basically look at we extract DNA, we do PCR. In this case, we looked at a potential pathogen called Bartonella. It's a mild potential pathogen, unlike viruses, which are very dramatic. It causes something called cat scratch fever. You get these rashes, and it's actually very common in forest-associated communities, but it's never diagnosed. So this is an example of a potential spillover. Oh, it started vibrating for some reason. And basically, something which is usually probably undetected, right? Another very good example of this is Q fever or scrub typhus. Again, very often found in forest-associated communities, almost never diagnosed because usually it goes away. It's not really serious, a fever for a few days, right? So basically, in terms of the rodent community, that is, we have various habitats here. Like I mentioned, we have grassland, we have forest, we have built-up area, the labor lines where people are working. And what we see is that this is just showing you which species. These are just species of small mammals. I mean, these are rats, for example. These are mice, etc. You can see that, for example, when you look at community structuring, grassland communities are different. So the grassland is GL. Are different from forest communities, are different from built-up communities. Okay, so basically, not just are there different species of rodents, say mice prefer to live in grasslands. This is a better way to visualize it. Basically, there are forests, there are grasslands, and there's built-up areas. Rattus rattus is our commensal black rat, right? And we have Rattus satare. It's an endemic rat of the Western Ghats. But you can see there are what we call shuttle species, these species which co-occur in different habitats. And these guys and their ectoparasites will be the one which can potentially cause spillover or movement of microbes in that community and landscape, right? So now the question is, how do we test this? So far, it's ecological data showing that there is this community. Some species are more common in certain locations. But what we can then do is, of course, just to show you some nice pictures, these are ectoparasites. And we know very little about, they're not all insects actually, some of them are not. But basically mites, ticks, fleas, we can't even classify them, but we do sample them. And we are assuming they're the vectors. So we look at the Bartonella from the ectoparasites, from the animals, and build a phylogenetic tree to see, are specific ectoparasites associated with specific species, and that is reflected through the Bartonella or not. So first of all, in terms of prevalence, overall the prevalence is really, really high. So in the community, that is 60% of the small mammals here have Bartonella, right? And in fact, some of them have really high prevalence. For example, Rattus satare and this CF fernandoni, we'll get to that in a bit. Don't have to focus on the names. But when we look at Bartonella prevalence overall, it is dependent on ectoparasite load. So that suggests that it is these ectoparasites that are transmitting the Bartonella. Now, whether this is pathogenic or not, we'll get to that in a minute. But if we also try to, again, reconstruct the origins of the Bartonella associated with these different species, you see, for example, overall, yes, you see specific Bartonella only occurring in, say, Rattus satare, but you also see an example here of a host switch, where a Bartonella from Rattus satare has moved to this Fernandoni mouse, okay? And remember, the Fernandoni mouse was part of the switch, part of the species which could shuttle or transmit things because it was in multiple habitats. So here's the phylogeny just to show you the lineages in brown are known pathogenic Bartonella, known to be pathogenic to humans. We don't know anything about pathogenicity in animals. And you can see that many of our Bartonella strains are very close to pathogenic strains. So there is potential that the Bartonella we have sampled in these small mammals are pathogenic. We have not tested it. That would require culturing the bacteria, infecting cells, and so on, which we have not done. And also, basically the ectoparasites carry the same Bartonella strain as the animal they were on. So that shows that it's basically is being transmitted by these ectoparasites, and it's not some strange, funky thing that's going on. Okay, so to summarize, basically Indian mammals are primarily Southeast Asian in origin, and we tested this with both macroecological and biogeographic and phylogenetic analyses. And mammal communities host many potential pathogens, sometimes with very high prevalence. For example, though, when we look at scrub typhus, which is caused by another bacteria, it's not as high in prevalence. And so these disease ecology studies can be quite difficult because you require many, many, many samples to actually get at levels of prevalence. So that's why, again, it's vibrating, but that's why a lot of people focus on something like Bartonella. And for example, Robin's lab works on avian malaria because the prevalence is quite high. And it's easier to do science when the prevalence is high. If, for example, for a species, for a potential pathogen prevalence is really low, it becomes really difficult. Okay. So now I'll just touch a little bit on some work we are doing, and clearly I'm going to finish very early. I'm sorry about that. But basically, some time ago, I worked on a very student-led idea of a bat harvest. So in a place called Mimi, which is very close to the border in Nagaland, close to the border with Myanmar, there's a community called the Bomar, and every year they harvest bats from specific caves. So basically they smoke caves, and then the bats fly out, and they kill the bats, and then they are exchanged as gifts as a tradition, and this happens year after year. So we thought this would be an interesting interface to look at this potential of spillover, because it's a high contact interface, right? There's a very high contact between humans and when people get bitten by these bats, and so on and so forth. And as we know, bats carry a lot of potential pathogens. They have really amazing immune systems. They're immunosuppressed, and this is suggested as one of the reasons why they live so long, because they don't have like immune reactions to pathogens, especially intracellular pathogens. They're not so good at dealing with extracellular pathogens. So basically, we sampled these bats and these humans, and we actually looked at serology to look at whether there's shared serological response between bats and humans. And we found shared serological response for several filoviruses. So filoviruses are, the famous filovirus is Ebola, right? But this is very surprising because there's no Ebola, there's no filovirus actually known from Asia at that point. They're only known from Africa. And so this study caused quite some commotion, and there were several other things which happened. But overall, it also helped me realize with a jolt that, while we're talking in the Western Ghats about 10 species or something like that, in the Northeast, there are many, many more species. If you remember that figure of, the diversity, first of all, 60% of our diversity is from, Southeast Asia, which is the gateway for that is the Northeast, right? And also, basically, there's a lot more species there. So if you remember the plot, and if you, basically 2/3 of India's mammals are in the Northeast, okay? So most of India's diversity is actually tilted towards the Northeast. So what we've done recently is we've actually started like a large program, and we just started to actually try to look at not just wildlife, but also livestock and humans. So a sampling site in this case is, caves with bats, forests with bats associated with the caves, villages associated with those forests and those caves, and their agriculture and jhum. They have shifting culture, shifting burn and slash and burn and shifting agriculture. So those sites, and the idea is to basically sample, this is just like, there's lots of institutions involved, but basically to sample these species for associated viruses and bacteria, and to sample humans as well for the same. So the idea is then we can actually look at not just what are all the viruses and bacteria in the bats, in the wildlife, in the livestock, but also in humans. And hopefully, just to show you, these are some of the habitats, extremely difficult fieldwork. A lot of the caves are at the base of these cliffs, and a lot of this is Meghalaya. And basically, like I said, we just started fieldwork, so it's really fun. This is Vivek, who's done a lot of work on small mammals. This is one of our person in the field team. She's holding a bat here, you can see. And you have to be very careful with these gloves and wear masks because, like I said, bats have many potential pathogens, right? And this is a small mammal called a niviventer. And we basically sample them, like holding like this for oral swabs, rectal swabs, wing punches, and so on. And the idea is hopefully to come up with some kind of a framework like this. So this is a very nice framework published a few years ago, which talks about risk. So you saw from the perspective of that community ecology, disease ecology study, it's very ecological, right? It says, okay, there's Bartonella in the system, there are these species, there are some species which have higher prevalence of Bartonella, some of those Bartonella are potentially pathogenic. So if, for example, we went to community health sites close by, like PHCs, and sampled people, maybe we could look at which of those are potentially pathogenic or spilling over to, say, the people who are working in the tea plantation, for example, right? But it's still very small scale. It doesn't really tell us much about, say, the risk of spillover more generally. So the idea here is to understand what's called hazard. And hazard is basically what bats are there, what rodents are there, what bacteria and viruses do they carry, how potentially pathogenic they are. So for example, I'm almost done. Basically, for example, coronavirus is not very pathogenic, whereas Ebola is very pathogenic, right? So like that, basically, what is their potential pathogenicity? But what's equally important is exposure. How are humans interacting with wildlife? When are they interacting with wildlife, right? And that we are trying to assess through surveys, and also looking at basically risk, which is the interface of these two through serology. So we are working together with Rahul Roy at IASc. He's developing some new multiplex serology, which can be done with really tiny amounts. In fact, we are actually collecting blood spots, so not even sera, which is what is usually used for serology. You have to have sera. And basically, we're trying to understand risk and how this risk varies across space. So the goal is to sample about 180 sites across Arunachal, Nagaland, and Meghalaya, and then to basically look at risk, calculate risk, and see, are there some places where there's more risk and others where there's lower risk, and then maybe design some kind of a way forward to think of how do we monitor this risk? Do we have to go and study every single bat, find out every single detail, or can we make some generalizations and educated guesses? So in general, basically we have some understanding communities is critical for biodiversity, disease ecology, and emerging infections. So the way we've always treated zoonoses is post hoc. There's an outbreak and then we rush to figure out who is patient 0, where did this spill over from, how did it happen? But it would be really nice if we could understand beforehand where are areas where there's a high risk of spillover, for example, right? And so this is what we're hoping to do. And as I said, I hope that came across, but while there's a lot of studies in the Western Ghats in general in ecology, a lot of times, we are missing, for example, the Northeast, which is really where all the action is. There's a lot of biodiversity there. And so we should be working in such landscapes, and also a lot of human-wildlife interactions, right? And so basically, fortunately, unfortunately, this cannot be done by AI. We have to do on-the-ground, field work, collect samples. Of course, analysis, AI can aid in analysis. There's going to be humongous data sets, but it will require boots on the ground sampling and fieldwork, something which is quite often disappearing from a lot of research now. There's a lot of data and people are just analyzing data. And so, that's probably a philosophical point, but I think this is also really important for the future. I have to thank Partha, who helped us in kind of securing this funding through DBT. So the work I presented was that of Ansil. He's really a fantastic, he was my PhD student. He's now doing a HFSP postdoc looking at bat coronavirus and immunity in Costa Rica. And this is Vivek. I presented some of his work as well. He was also a postdoc with me and now works with the MSc Wildlife Program. The phylogenetic analysis, the big murine phylogeny, was put together by Chetna, an undergraduate at IISc, and Aparna Lajmi, who was a PhD student at IISc but also worked with me briefly. Thank you so much for listening, and happy to answer questions.
Thank you. I'm sure there are lots of questions. Yes?
My clarification is in particular with your first part of the talk on biodiversity, where you mentioned that many of these species move from Southeast Asia or northern part or the western part. Over the years, when you make this phylogenetic tree, did you found that there was cross-mating in these species, and you found some new species which are being evolving while they are within the country?
So that's a very interesting point. Actually, that would recently, in general, there has been many discoveries of hybrid speciation, where two species hybridize and create a new species. Generally, not at all common in mammals, much more common in plants, for example. We have not found any evidence of that. It really seems oddly that India is kind of empty of there's not that many endemic species in India. So maybe that was because there were a lot of extinctions. We don't know what that is because of. But one of the things is that, Southeast Asia is extremely wet. So there's a lot of historical contingency. That is, species that have evolved in wet environments tend to stay in wet environments, right? Whereas species which are from the west are more arid, dry zone species. They tend to stay in more drier environments and so on. The Himalayan guys never get past the Himalayas, right? They're used to more cold environments. So in India, there's a lot of gradients driven by lack of rainfall. And that kind of prevents probably some species even from meeting each other, as you would say. But communities are dominated by Southeast Asian. Communities and numbers are dominated by Southeast Asian species.
My question is that when India broke off from the African continent, is it that there was nothing, no
There
mammals,
were some species, and
so
those
what
are
was that fraction
right.
that still persists to this day?
Not mammals. So there were, because mammals evolved
Much later.
After that, while it was floating. But the classic example is the purple frog, Nasica batrachus, which is basically a Gondwanan species, when India was part of that larger landmass. But for mammals and birds, most of the speciation happened after KT, 65 million years ago. They were around, but they started speciating after that. And so India kind of missed out. There was also a lot of volcanic activity on India as it was floating. So, yeah.
So this is just a continuation, actually, of his question. So are there species that have evolved in peninsular India, which we can say are just...
There are some, but
very few.
Very few.
And this is true for across, mammals?
No, for mammals, birds, for example, robin has found some endemic radiations, but generally very, very few.
The Himalayas have risen over the last 50 million years.
Yes.
And when they had touched Tibet, there was some migration from the north, particularly of mammals.
Yes.
And as it began rising, there might be an evolutionary sequence of processes
definitely.
that distinguish them, between what exists in the foothills and...
Right. So from what we have studied, I didn't present it, we have done some biogeographic work in the Himalayas that suggests more patterns of dispersal. So basically the Himalaya is like this. The eastern Himalaya is more tropical and the western Himalaya is more temperate. So we see patterns of dispersal and a little bit of speciation within, but not intense speciation in the Himalayas. So it's more a pattern of dispersal, but we're going to be sampling Arunachal, Nagaland, Meghalaya. Hopefully we'll get more. We need to do more sampling. We really don't even know the species of rodents in the Himalaya, very frankly. It sounds kind of silly, but that's how it is.
Here.
Okay. Just a quick follow-up question on the geoecology.
Yeah.
Madagascar and Western Ghats.
There are some shared species.
The flora fauna must be, the biodiversity must be. No?
No.
There's no.
families and
of
some
trees.
Yeah, it's been studied.
Yes.
Carefully.
That's in fact one of the proofs of plate tectonics
Right.
is, the fact that there are shared species across these very currently disparate continents. But definitely mammals.
This was the breakaway of Palakkad Gap that created Madagascar. So
No.
that's the popular version.
No.
That the Palakkad Gap was actually created by the breakaway of Madagascar.
No,
No?
I don't think so.
That's
Here.
Yeah, hi.
If the mammalian evolution more or less happened after India broke off from Africa, then
globally.
how come you have similar species like hyena, like rhino, like elephants?
Those are all dispersed, yes. So for example, rhinos are very, very widely distributed, right? And they are larger. So we're talking about small mammals. Their ability to disperse is much lower. So their distributions in general are smaller. But these species, you're right, hyenas, crazy wide distribution, right? Hyenas, wolves, leopards, these are larger species and they have potentially dispersed. But definitely there are still patterns. Like tigers are Asian, they've evolved in Asia, most probably South China, whereas lions have evolved in Africa and have dispersed. So phylogenies, phylogeography, that is looking at patterns of genetic variation in populations across space, can allow you to infer those patterns of dispersal, the directionality and timing.
We have 12 seconds for one very quick question.
Raghavan.
So many of the Nipah outbreaks are on the western coast, not in the northeast.
Yes. Yeah. So basically, the Western Ghats, so that map, I just showed it, the determinants of high probability of zoonoses is high mammal biodiversity, high livestock density, high human density. And the Western Ghats is a biodiversity hotspot with the highest human population density in the world. So yes, definitely there is zoonoses in the Western Ghats as well. But I think, I like being an explorer a little bit, and the Northeast is really very, very unknown. A lot of data from China, and so that's nice to compare, actually. A lot of similar species. So hopefully we'll find some interesting things.
Well,
Hi.
Hi.
Yeah. So very nice talk. I just had a very quick question. So the local populations which are in close proximity to the wildlife there.
Yes.
Over the years, or maybe over the last, how many years they are together, they might have grown immune to some of these pathogens. Do you think some of these things might be different for the mainland or maybe other parts of the world? And how are you going, is it something people can potentially study to see that maybe the locals are not something who might really be affected
people,
badly?
you mean? Local
yes,
people.
Yes.
Yeah, so basically that is probably very true. There's a lot of signatures of in the genome of humans of adaptation to, for example, malaria and so on. Unfortunately, in this project, we didn't get the funding and permissions to look at that for humans. Maybe it's something we can do later. There's a lot of biology also, for example, understanding the pathogens and so on. But yes, that's a really important point. Yeah. Thank you.
Okay.
Thank you so much.
Thank you, Uma.